The overall goal of this procedure is to introduce undergraduate science students to microarray technology by examining the expression differences in yeast undergoing oxidative stress. This is accomplished by first isolating total RNA from yeast cultures treated with hydrogen peroxide and the untreated controls. The second step of the procedure is to generate and purify cDNA from these RNA samples. The cDNA is then used to prepare biotin-labeled cRNA via in vitro transcription. The final step of the procedure is fragmentation of the biotin-labeled cRNAs for hybridization to the Affymetrix yeast gene chips. Ultimately, results can be obtained that show the expression differences in the two yeast samples, and through bioinformatics demonstrate the power of microarray analysis to undergraduate students.
The Vermont Genetics Network outreach team first had the idea for this method when they were charged with delivering microarray technology to science students throughout the state of Vermont in order to enhance their science education. To date, this method has been delivered to multiple sites at 8 baccalaureate colleges throughout the state.
So the main advantage of using microarray of a teaching module is it gives us the ability to teach general molecular biology techniques that are used every day throughout the lab to a group of individuals. And we also get to use this to teach to students and teachers alike. In our groups, we actually have students and professors working together on the same project. They learn certain techniques that take a lot of finesse, like handling RNA, which is not an easy task. They learn how to precipitate DNA using visualization reagents. They actually get to use the techniques that they're going to encounter in graduate studies or in everyday molecular biology labs.
Though this method is suitable for gaining insight into oxidative stress in yeast, it's certainly applicable to other model organisms and other biological systems that you may want to look at gene expression changes, whether they're associated with developmental changes, environmental toxins, specific disease states, and really many more.
To examine the expression differences in yeast undergoing oxidative stress, total RNA is first extracted from 2 yeast cultures by enzymatic lysis. One culture was exposed to oxidative stress by a 1-hour treatment with 0.5 millimolar hydrogen peroxide in Hanks buffered saline, while the other culture was treated with only HBSS as a control. Begin by labeling 2 1.7 milliliter microcentrifuge tubes with the appropriate identification information. Transfer 1.5 milliliters of the appropriate yeast culture into each tube. Centrifuge the tubes at 5,000 Gs for 2 minutes at room temperature. After centrifugation, use a micropipette to carefully remove and discard the supernatant without disturbing the pellet. Check that the pellet is big enough before continuing with the procedure. If the pellet is too small, add 1.5 milliliters of culture into the same tube containing the pellet and then centrifuge to obtain a bigger pellet. Discard the supernatant with a micropipette, removing as much of the liquid as possible from the yeast pellet. Then add 100 microliters of SG buffer and 30 microliters of lidocase solution to the yeast pellet. Vortex to mix. Incubate both tubes for 30 minutes at room temperature. During the incubation, gently swirl each tube every 10 minutes to generate spheroplasts.
One of the most critical steps when you're doing spheroplasting of yeast is to make sure that you actually created 100% spheroplast after the treatment. That means not all lytic enzyme is created equal, so you have to check to make sure good lytic enzyme is giving you good spheroplasting. Meaning you have to take these samples, put them onto a microscope slide, and check under the microscope by adding 0.1% SDS to ensure that you do have protoplast formation.
To observe complete spheroplasting, pipette 10 microliters of the yeast sample onto a microscope slide. While examining the sample under the microscope, add 1 microliter of 0.1% SDS to cause the yeast cells to swell and form perfect spheres or spheroids. It is important to observe that the budding cells form spheroids as well. If partial spheroplasting is observed, an extended treatment with lidocase is recommended. Once complete spheroplasting has been confirmed, add 350 microliters of BRLT buffer to each tube. Then add 250 microliters of 100% ethanol. Ensure the tube is tightly capped by holding the lid closed and vortex vigorously for 1 minute. This procedure will lyse the spheroplasts. After this, use RNeasy spin columns to collect and clean the RNA from the 2 cultures. Finally, evaluate the quality of the extracted RNA using a 1.2% precast agarose gel for electrophoresis. To prepare biotin-labeled cRNA, the total RNA extracted from the yeast cells is first used to synthesize cDNA by reverse transcription. After cDNA generation, prepare phase-lock gel tubes for use in cDNA precipitation. Centrifuge phase-lock gel tubes at full speed for 1 minute to ensure the gel is at the bottom of the tube. Do not vortex phase-lock tubes. To precipitate cDNA, pipette 162 microliters of the bottom layer of a pH 8.0 tris-buffered phenyl chloroform isoamyl alcohol, or PCI, mixture, and add to the 162 microliter contents of the second-strand cDNA synthesis reaction tube. Equal volumes of aqueous and organic mixtures are required for this step. Vortex for 5 seconds to mix the contents. Next, use a micropipette to transfer all of the cDNA PCI mixture to the phase-lock gel tube. Do not vortex the phase-lock gel tube. Centrifuge at full speed for 2 minutes. Then use a micropipette to transfer the top layer from the phase-lock gel tube to a newly labeled 1.7 milliliter tube. Try to collect as much of the layer as possible. Add the following to the 1.7 milliliter microcentrifuge tube: 405 microliters of 100% ethanol, 80 microliters of ammonium acetate, and 1 microliter of pellet paint. Vortex briefly. Place the tube in the centrifuge with the hinge of the tube facing out and centrifuge at full speed for 20 minutes at room temperature. When centrifugation is complete, gently remove the tube from the centrifuge, being careful not to disturb the cDNA pellet. The pellet should be pink because of the pellet paint and approximately the size of a grain of salt and on the side of the tube under the hinge. Place the cDNA pellet on ice and proceed immediately to cleaning the pellet. To begin the procedure for cleaning the cDNA pellet, use a P1000 micropipette to carefully remove all of the supernatant from the tube. Be careful not to disturb the pellet, it is your sample. Add 500 microliters of cold 80% ethanol to the tube. Gently cap the tube and invert it slowly several times. Watch your pellet very closely to make sure it does not become detached from the side of the tube. If the pellet becomes detached, you can get it back to the bottom of the tube by either placing the tube back in the rack and letting the pellet settle to the bottom, or centrifuging the tube at full speed for 15 seconds. Next, use a P1000 micropipette to carefully remove the ethanol without disturbing the pellet. Tip the tube to enable removal of as much liquid as possible. Add a new aliquot of cold 80% ethanol, cap the tube, and invert it slowly several times. After removing all of the ethanol possible by using a P1000 micropipette, centrifuge the tube at full speed for 5 seconds. Then use a P10 micropipette to remove the last few microliters of ethanol without disturbing the pellet. Place the open tube in a drying box for 10 to 20 minutes to evaporate the remaining ethanol. The pellet is easily lost once it is dry, so be careful to handle the tube gently and to close the cap when drying is done. Visualize the dried pellet to confirm it is present in the tube. Finally, resuspend the dried pellet in 22 microliters of RNase-free water and place the tube on ice. This cDNA is then used to prepare biotin-labeled cRNA by in vitro transcription using the Enzo bioarray kit. After the biotin-labeled cRNA has been cleaned and quantified, it is fragmented for target preparation.
Once the cDNA has been generated, we then use standard in vitro transcription methodology in order to generate biotin-labeled cRNA. The cRNA needs to go through fragmentation before it can be applied to the microarray chip.
To begin this procedure, transfer an amount equivalent to 5 micrograms of cRNA to a labeled 0.5 milliliter PCR tube. Add enough RNase-free water to bring the total volume to 16 microliters, and then add 4 microliters of 5x fragmentation buffer to the tube. The total volume in the tube should be 20 microliters. Vortex the tube and centrifuge for 10 seconds. Then incubate the tube at 94 degrees Celsius for 30 minutes in a thermocycler. Put the tube on ice following the incubation. The fragmented and unfragmented cRNA from each sample is then assessed by electrophoresis using a 1.2% precast agarose gel. When the run is complete, the gel is visualized on a transilluminator and an image is acquired. The final synthesis product is hybridized to the Affymetrix yeast gene chips, and representative results from the microarray analysis are shown here. This first figure is an example of a scanned Affymetrix yeast gene chip image generated by the Affymetrix gene chip operating software. This is a 2D scatter plot of all genetic transcripts, about 6,700 genes, comparing control and treated yeast data. Each point represents a single gene. Genes colored in purple indicate genes that are differentially expressed, while genes colored in red are not. In this example, most of the differentially expressed genes are those involved in cell cycle control, as indicated by their descriptions. This flowchart from the Database for Annotation, Visualization, and Integrated Discovery illustrates differentially expressed genes in an affected biological pathway. The genes indicated with a red star indicate the downregulated genes in the meiotic pathway. Finally, representative results of a volcano plot generated using GeoSpice's Gene Sifter software are shown here. Control and treated samples were compared with a p-value cutoff of 0.05 and a 1.5-fold expression change cutoff.
We chose using yeast because yeast are easy to grow quickly, but we also realized the most critical part of working with yeast is actually creating decent spheroplasting. One of the things in microarray we don't want to do is actually do an incomplete digestion and actually only spheroplast cells that are in a G1 or a G2M phase, and then you're doing a microarray experiment on yeast that are in a certain replication phase. Another complicated point that we try to bring home in this exercise is that pelleting cDNA people talk about every day, but it's not necessarily easy. And what we've done in our module is we've actually used specialized tubes and visualization reagents so we can actually spin down DNA and you can visualize a pellet, which makes it easier for students and teachers alike. So that can be used in all phases of DNA and RNA work.
After its development, this module has really paved the way for faculty at the baccalaureate institutes to explore possibly looking at other model organisms or other treatment regimens that may be compatible with existing curricula or possibly their own research interests. And to give you an example of some of the modifications that were made, there was one site that had looked at the effects of herbicide treatment on yeast, or another that looked at lampricide treatment in yeast, while another site looked at the developmental changes in a mammalian cell line that they were particularly interested in. And lastly, another site had looked at the impact of differential light sources on Arabidopsis or plants.
After you watch this video, you should have a good understanding of how to set up a microarray experiment with undergraduate science students, including the isolation of total RNA from yeast, the generation of cDNA, and the fragmentation of biotin-labeled cRNA. Hands-on experiences with such high-level technologies helps to reinforce and strengthen undergraduate science education.